Yeah. Good evening, all, and thank you for joining timely.
Warm good evening.
Yeah. Yeah. Is it my voice is clear to everybody and screen is visible?
Yes, yes.
Yes. Yeah. Great.
Uh,
Yeah.
it's
Yeah.
clear.
So, yeah, please. Fine. So, today's session will be an overview for the coming 3 months. What we are going to discuss. So, what is our overview? Already shared the syllabus, but even though how it is linked with our routine sessions, I will going to explain here. So today's session, as I posted the agenda already in the WhatsApp group, so same topics will be going to discuss in the presentation topic. At the meanwhile, we are going to refer the guidelines as well. So how biosimilar guidelines, what are the expectations, and how during our development we what we had to refer. So these are all the stages. So not only in the development, not only in the manufacturing. So complete model 3, the quality data requirement, and as well as the clinical requirement as per safety guidelines, as per efficacy guidelines. So what is the preclinical and clinical requirement? So these all will be referred. At the same time, at the ultimate time, we targeting to know the regulatory strategies and regulatory filing pathways. That is the main scope for the people who joined here. So there is a CTD organization structure. So what we are going to discuss today in individual documentation practices, when it is reaching to the regulatory department, they will transcribe the data into the dossier format. So that is the ultimate job to know everyone who has been worked on the different departments or want to learn the regulatory write-ups and dossier development that we are going to learn coming 3 months. In this 3 months, so first 40 sessions we are going to discuss on the technical sessions, which is required for the dossier development. After completion of the technical sessions with related to cell banks, related to upstream process, related to downstream process, complete drug substance strategies, and followed by drug product formulation strategies, analytical strategies, QMS, quality management system strategies, how it is linked with our respect to requirement of the filing activities. Some may be required for the filing activities, some may require for the dossier development, some may require for the dossier part of submissions, some documents is required for the part of audits and visits. So like this, every GMP documents and every non-GMP documents may require for the part of submission, may require for the dossier development. So how it is linked with from initial stage to final stage, that will be linked every day, every session. Today's session linked with tomorrow session linked with day after tomorrow. So like that. So there is a link up. So
Sorry
if
to
you miss,
interrupt. Sorry to interrupt.
yeah, please.
Even GLP document also required. No, GLP and GCP documents.
Required. That's what I'm saying. Non-GMP. When you consider as a GLP, QC documents, all things is required. Yeah, so which is supporting directly or indirectly for the part of your submissions. So maybe part of preparation of the any GMP documents or non-GMP documents. Those are required. As and when required, the document has to be open and to be shown or to be part of attached with along with your those here or maybe part of GMP activities required. Yeah. So that is what we are going to learn today. Yeah. So entering into our presentation topic. So before going to the presentation, I will show that guideline how it is linked with our activities. Before entering to the one of the biosimilar guidelines, we had to refer it. Is guideline is visible? Yeah, is screen visible?
Yes,
Yeah.
yeah.
Yeah. So what is mentioned? This is the European guideline. So no worry, we'll go with every day it is a link up with the biosimilar guidelines especially. So if you see here, the guideline what is mentioned, guideline on similar biologics medicinal products. So to file a similar, that means similar means biosimilars only, or we can call it a similar biologics, either biosimilars or similar biologics. So there will be terminology will be different, but looks like same only. So which is already registered, a biological product already registered in their market. Against that, we are going to develop a biosimilar product to register against this biological product. So this biological product registration when is done and it is having a some patent protection. So once that patent protection is complete, then we'll go for filing of this one. So during our development of this similar biological product, we can go for preclinical, we can go for clinical, and we can go for submission as well. But authority will review and complete the process, even though every successful review and every successful submissions. So they will not give permission to marketing authorization. So once this biological product get complete their patent protection, patent exclusivities get complete, then we'll get the marketing authorization permissions. So license will get after this product is expired the patent. So to develop this product, so the difficulty is to read the guidelines. It will take line by line. It will be made difference. By showing the guideline, I will explain how we can learn from these guidelines. Because if you go line by line reading, it's like in newspaper. It's not correct the way of reading. But I will explain the oral so that it will be catch the direct requirement. So if you see in this introduction part, in this smallest guideline, a regulatory framework. So what is the regulatory framework? What is the scope? Application of the biosimilar approach. How to approach to the put application. And choice of reference product. How to choose the reference product. A principle of establishing biosimilarity. A simple 3 to 6 pages, just for 3 to 4 pages of document will give you a lot of knowledge today. In this case, a regulatory framework means, so when you're telling that a company may choose development of biological medicine product claim it to be similar to be the reference medicinal product which has been granted marketing authorization in the European area. So this is the first thing required for the your submissions and development. Initial development is itself is required, and submission point of view also we have to refer the reference guidelines. So when you're going for a reference product, when you're going to use for the granted, which is approved product in the EU region. So for the part of protein characterization, part of biosimilar assessment. So when you're talking about part of biosimilar assessment, there will be three different of assessment will be there. One is analytical similarity assessment with the quality module. So where we are going to do comparative lot studies. So that means our in-house product versus the innovator product, which is registered in the EU region. We have to procure that lot and we have to lot comparison, head-to-head comparison. 3 lots innovator product, 3 lots our in-house product. We are going to compare initial before going to preclinical approval itself, 3 plus 3 lots with R&D batches, development and optimization completion. We had to be compared. In that comparability studies needed to generate evidence of substanting a similar nature in terms of quality, safety, efficacy. So quality will be ensured by quality parameters, analytical similarity assessment. When it coming to the safety parameters, preclinical studies, we are going to ensure it. And efficacy point of view, part of your phase 3 studies. So everywhere, quality part also you are comparing with innovator product and our product. Safety point of view also, we are going to compare with innovator product, our product. And efficacy point of view also, we are going to compare with innovator product as well as our product. So here, the quality comparison. What is the purity of innovator product? What is of in-house product purity? What is the potency? What is the this product potency? When you're going to compare even though stability compared to accelerated stability also required to compare it to generate lot of data to the evidence documents to be provided for the part of submissions. Like this is the regulatory framework when you're looking for the European market. So now coming, so anyhow, need not to worry when you're going for the filing activity. Once you complete the technical sessions, when you're going for the filing activity, if you remember the day 5 free sessions, I shown this type of anecdotes. I shown this type of numbers for the registration purpose, where we refer that variation filing. Same thing will come for the part of submissions. So what is the approach for the entering into the medicinal products for human use in generally we call committee for medicinal product. So this is the path for the filing of our biological products for the human use. In generally, CHOMP we will call as a CHOMP. So how to enter in here, we'll going forward, we'll be going to learn in our sessions. Just I'm rolling it on. So what is containing of this document? Legal basis. So nothing but patent related one and related to commitments, agreements. This is a part of once product is going to register. So we have to do agreements and legalizations. Those and all will be part of it. Then general principles. So what is the principal application of the per biosimilars approach? So should be referred. A biosimilar product, medicinal product contains a version of the active substance of the already authorized original biological product. This is the main target. So which is already authorized in the EU region, that product has to be taken as a reference product. So very clearly mentioned here what is required. So what is the comparable guidelines is mentioned as per ICS Q5E, and we'll refer going for all ICS guidelines, how it is linked with that. So like this guideline can help us to understand the biological products. So now choice of the reference product, which choice I have to choose it? How many lots we can choose it? In case of 3 products, 3 lots can be choose from EU, 3 lots can be from US FDA also, and MHR also can choose it. If that product is not registered in the EU, but we are going for filing to the EU, but we can go for US FDA approved product as well. We can use a reference product. That is the choice of product is going to explain here. Then principles of establishment biosimilarity. as I mentioned, quality and analytical, preclinical and clinical. These are the 3 stages required for the establishment of biosimilarity. Biosimilarity means 3 phases: analytical similarity, preclinical similarity, clinical similarity. In these 3 phases, we have to show the evidences. So that is part of our regular sessions will be covered. So this is what, if you want to file for the EU, we have to refer like this guideline. In case we are looking for the US, simple way, open the US guidelines and refer the US requirement. When you're going for the MHRA market, then simply refer the MHRA requirement of the filing activities. Accordingly, their pathways, regulatory framework, we have to look into the reference guideline. Similarly, India market, also there is a guideline which is produced in 2016 by our biosimilar guidelines. So that is also required. Even though I shown in the during our session 4, pre-demo session 4, so there was shown how to refer that guideline, what is the expectation. Every country, the expectation is the product should be registered in their own country. At the same time, this product should be either registered in the EU region or maybe ICH regions in generally. So it's not possible to get it from every country for every country submission. We can choose ICS regions of the product registration status. We can sorry, Ramesh Patil, please. Okay. So we have to refer like this guidelines. So when we are going back to the our presentation so that we can understand more today. Is it clear to everybody how to refer the guideline?
Yeah.
Yeah. So now this slide I given a presentation even the demo session, but even though we'll refer for some time so that it will give more overview on the how product development will happen. So as we know, discovery phase is nothing but R&D phase, where we are talking about cell banks. We're going to talk about upstream, downstream, entire R&D activities. So complete optimization of the process, including analytical part. So we'll be developed here. And before going to initiation of R&D activity, we should get the permission from the local agency, local authority. So there is a test license is required. At the same time, import-export licenses required. This is the local MOH requirement to procure something from outside of the country, outside of the state to inside of the our country, because we are going to dealing with any recombinant genetically modified organisms. This should, so before going to procure them, we will have a IBSC meeting in case of India. So then that meeting, we are going to produce our product which we are going to propose for the development. So what is the indication? What is the purpose? How we demand in the market? How it is required? And will be taken IBSC members in that meeting will be get approval. Then we'll go for applying a application process for the test license as well as import and export licenses. Once you got the import and export licenses as well as test license, will grant permission for the to conduct R&D studies. So once you receive this granted R&D permissions, then we'll go for development, research and development, and actually will start development and will be optimize our biosimilar products. Even the biological products, biosimilar products can be start the actually research and development will make the final optimization with respect to analytical, with respect to process. Once these both are optimized, we will go for consistency batches. These consistency batches, we will see the evidential document generation, protocol-based execution, and report generation. In this batches should show repeatability, reproducibility, and consistency. So these batches can be used for preclinical studies, and these batches can be used for stability studies of the PCT stage or small R&D stage, and these batches can be used for the comparability studies, protein characterization. Like this, in biological products, many activities will be done for those products. So the product related to DS, drug substance, same drug substance can be used for the drug product formulations. So that there is a DS, there is a DP. So once this batches is placed with respect to activities, so then we'll look for the strategy to go for preclinical studies. So to conduct preclinical studies, we have to raise a protocol to submit for the agency. So to get a permission from local authority. So R&D and your respect to preclinical study should be taken care by their own country, local authority. And going for clinical trials, that is choice of manufacturers, choice of the sponsor organization, where they can go for country-specific clinical trials or region-specific clinical trials or any global clinical trials. So here, when you put the application here, protocol. So I shown in the day 4 session, anyhow, going forward, preclinical application, we'll see what are the data requirement. So the data related to what I mentioned here during development and optimization. So those all data will be cumulatively going to attach your protocol. The agency will review the data related to similarity assessment, main thing, protein characterization, consistency report, stability report, photo stability, there are the reported degradation stability studies. These many things will be reviewed and agency will be accepted for the go for preclinical studies. So we had to tie up with any CROs and get the permissions from agencies and start tie up with any CROs, go for animal studies. So since this is are the biologicals and biosimilars, when you go for biosimilars, there will be somewhat exclusivity there, but required to conduct safety studies to know that any immunogenicity reactions are there. So after comparative studies, conducting of immunogenicity studies, we'll get the reports. That reports will be enclosing and going to submit to the web-based portal submissions to the local MOH. So the local MOH will review the reports of preclinical studies. Then they will grant the permission to conduct clinical trials. Here, the sponsor organization to be taken a decision based on their business strategy, based on their capacity of the organization funds. They can choose either local with respect to their country clinical trials, or maybe any individual other countries, or maybe international clinical, global clinical trials, or any regional specific clinical trials. So in that case, if you want to file EU, or you want to file USFDA, or you want to file MHRA, so even though you want to first time registration, you are going. So already product is registered, already being submitted in the some other countries. Scientific evaluation, and we'll take the scientific advice meeting. So here the team will be assess our complete product already being submitted, being already supplying the other markets, looking for the FDA, looking for the EU. They will assess and let us know that where additional clinical trials is required or not required. Or this product is very new, it is a new biosimilar product going for the new registration in the for clinical trials. So there is a pre-IND meeting in case of USFDA. In the pre-IND meeting, in IND means investigational new drug. In that pre-IND meeting, instead of give permission go for clinical, and there is insufficient data from this preclinical phase or in the R&D data, there may be choice of any challenges. For that reason, they will conduct pre-IND meeting. In this pre-IND meeting, they will ensure any previous human exposure is there, already existing molecule in the market, or related to preclinical data, related to R&D data, CMC data, they will verify. After assessment of verification and having a pre-IND meeting with sponsor as well as any consultants with respect to FDA region people, then they will give the permission to go for clinical trials. So let's imagine either you or maybe you're going for the FDA. So we'll choose either region specific, country specific, or global clinical trials. We will go for phase 1, phase 2. If phase 2 is applicable in the vaccines or any new biologics, in case of biosimilars, not required. That is already we're going for, we'll see how it is the strategy will be there. Then we'll conduct phase 3 clinical trials. Then we'll collect the reports and we'll go for completely those are preparation of module 1 to module 5. So finally, we are going to the new drug application with respect to which region or which country you are going looking for the registration. So when you submit the data of module 1 to module 5, which is containing module 1 administrative information, module 2 will be index for the process quality as well as preclinical and clinical data, and module 3 is the quality, module 4 is the safety, module 5 is the respect to clinical efficacy data. So when you file a dossier here, marketing authorization or biological license application, the agency will receive and they will acknowledge it, and they will start review process. This review process, which is you will be different, but ultimate task is to review and give the queries and ensure the plant GMP status and go for permissions. And EU status will be different, and USFDA process will be different, but ultimately the process will be remain same, but the number of days or review process will be somewhat different. So they will review and they will come back to the plant GMP, and plant is ensure that in line with their GMP requirements. So they will issue the GMP certification, and related to submitted dossier, is there any gaps? Is there any insufficient data? They will raise a queries and they will give the certain timelines, either 2 months, 3 months, based on the type of query. So in that period, we had to address the queries and we had to complete the plant GMP certification. So once everything is successful, they will issue the marketing authorization. So once marketing authorization is issued, then we'll go for process validation batches. So that is the reason we have been taking a class on the day 3 process validation, how it is linked up. So sometimes clinical batches itself, we can do process validation based on the strategy of the organizations, sponsorships, and based on the marketing, post-marketing authorization also, we can do process validation based on the any differentiation of site, manufacturing site, any differentiation of the batch size, differentiation of the equipments, different in the any process-led thing. We'll go for new process validation. So we'll produce the process validation with respect to drug substance against the drug product, and that will be tested in the validated QC methods, and will be produced the results, and we'll prepare a reports. Then final modified, those will be prepared, and meanwhile collect the stability, and meanwhile conduct the biosimilarity, meanwhile conduct the protein characterization, and will completely cumulate to final data will be put in the dossier. Then after showing that 6 months of stability reports, we will get a WHO GMP certificate where we can export from our home country to other countries. Then we'll launch the product in the market. So once you launch the product in the market, so it will reach to the respective hospitals, respective medical stores, and where will be required to consumers. So once it reach to the market, and doctors will be written the prescription to take this product for the either treatment or maybe whatever the requirement. So the people will start, the patient will be start utilization, and there is a requirement of phase 4 in clinical. Before approval, we have been conducted clinical trials, phase 1, phase 2, phase 3. Phase 2 where is a applicable. Phase 3, phase 4 is a post approval marketing authorization. So this is a surveillance, post approval marketing authorization, surveillance data going to collect in the real patients. So we are going to collect, study long-term effectiveness and any side effects, any adverse events, anything happen to the patients. So we have to be collective information for certain duration, like 3 years, 4 years, we have to collect it. That data has to be again to be placed in the dossier. When your product is going to approve here, you having only phase 1, phase 2, phase 3 data. So after approval, launching the product in the market and utilization of the people, going forward, we are going to keep on add on the service. So every year, going to collect it and placing in the dossier. So it is a part of annual submission required to report it. In that annual submissions, there is no any reported adverse events. There is no any related to issues. Anything need to be intimate the agencies. So if there is any adverse events, there is a chance to hold the manufacturing or recall the product, or maybe stop the manufacturing. Anything will be warnings will be coming to the organization. That is what during conducting of preclinical trials, clinical trials, we should not any manipulate the things. If it is happening anything like this and get the fake information and get the marketing information, later stage is going to spoil our organization. So that is what utmost care to be taken during clinical trials and preclinical trials, ensure the safety and efficacy. So it is everything successful here. We should not bother about here. So when you're going to make similar quality of the product of the biosimilar product, it will be reproduce same results in the during our commercial launch, commercial product. So like this required for your data collection. And meanwhile, in future, you want to revise the process, nothing but alternate raw material inclusion, or maybe changing in the capacity of the product, or changing in the facility, or addition of the additional facility. There is a called post-approval changes. So like this, in coming 3 months, we are going to learn about R&D process, going to learn about regarding manufacturing process, going to learning about how to prepare the dossiers, how to go for the filing activities. As I mentioned the syllabus, we are going to learn about how we are going for the variation filing. With respect to preclinical and clinical point of view, we are going to take a overview only. With our 40 sessions, we'll talk about complete CMC, and another 10 sessions for the dossier development, another 10 sessions for your filing activities. Like this, 60 sessions will be there. Each session 90 minutes, 16 to 90 minutes, 90 hours the course. Is it clear to everybody?
Yeah.
Yeah, fine. Now,
I wanted to ask one question.
Yeah, please.
Yes, with respect to the phase 4, would you submit annually the report from the phase 4 in your next dossier submission, or it will be considered during the product, the annual review of the product? How is it done? I didn't get that side clear, whether you submit it as part of the dossier submission.
I'll come back.
Okay.
What happened here? We got the marketing authorization with your respect to until phase 3 data, right? After submission of this, after approval, we are going to launch our product and going to giving to the agencies for the collection of the data. So there will be collection the clinical team from the hospitals where our product has been registered and where has been submitted supplying the patients. There we are going to records will be collected. How many quantities we produced, how much quantity has been consumed. From that, is there any reported adverse events? Anything is there? So that will over a period of time, we are going to collect in a year of time. End of the year, going to collect a phase 4 data. So annual submission. It is like a annual submission for the new products. Clear?
Okay. All right.
Yeah.
I just wanted to ask two questions, sir.
Yeah, please.
For anything, whether it is a new biologic or it is a biosimilar,
Hmm.
we have to approach IBSC for sure to begin with.
Definitely. Yeah,
Yeah.
because biologic it is a biosimilar. We should first put application for the test license point of view, and regarding preclinical application point of view, approach for the first thing is IBSC. That is the
Okay.
that is the responsibility of IBSC board to take care of biotechnology products.
Yeah. So while we approach IBSC, it is kind of an online portal, as you said that day. For
Online
other
portal.
activities, it is there.
There is an online portal application,
Okay.
and with respect to there is a meeting will happen. In that meeting, we have to be produced, and there will be annual meetings or like IBSC meeting will be there. That will discuss during our
Okay.
national application, preclinical application.
Yeah. And this will be the case also for US FDA or Europe related submissions, whatever we are coming up with for the export purpose. Also, it is a mandatory thing.
Yeah, until here, what are the products you are going to manufacture in India? Even though you are looking for the either FDA or EU regions, until this phase. So discovery phase and preclinical phase, the process should be in line with your Indian government requirement, Indian guidelines. Even though you are referring with FDA guidelines, even though you are referring with the ICC guidelines, but you should be in line with your Indian guideline requirement. That is what Schedule M, Schedule Y has to be referred for the part of development, part of our clinical and the preclinical things.
Yeah.
Right? Yeah. So this is the dossier. So where we can see module 1, module 2, module 3, module 4, module 5. So module 1 is a like, yeah, please, someone is asking something. Okay, fine.
My doubt is here.
Yeah, please.
Thank you for elaborated explanation. My doubt is there are two doubts regarding selection of RMP. Regarding selection of RMP, if the RMP is not at all available in the regulated or the non-regulated regulated market, if it is or to respect to regulated market, if it is available somewhere in the non-regulated market, then can we use that for this market? I mean, whatever.
It is not at all registered in the EU region or maybe in the US region. Then we can't consider it is a biosimilar. It will go with like a biological product, new innovator product. When the product, if the product is not registered in the approved,
if it is registered in Japan market or any other market, African Japan market, then can't we use that as a RMP for this? That is a new drug for this EMA market.
Even though we'll use as a reference product, but they will consider it should be registered in their regions first. That is the main requirement, is to consider as a biosimilar. It is not registered in the EU regions or maybe FDA regions, in the ICH regions. It should be at least one of the ICH region when you're going to file as a biosimilar product. Either it is a Japan or maybe FDA or maybe in the EU. If it is not registered in these three regions, or you are going for new filing into these markets, it can be considered as a new biologic or new innovator product. It's not a biosimilar product.
Okay. And next question is regarding ethnic data. Like which data will be like? Can we use mixed ethnic data, or the respective regulatory market data? Means country data is required with respect to safety, efficacy, and other data.
Yeah, that's what. So we should refer, when you're going to refer the ICH guidelines, as per mentioned that M4S, M4Q, and as well as M4E. During your strategy development, process development, when you refer M4Q, it is in line with all ICH guidelines. When you're going to refer M4E guidelines, efficacy guidelines, and M4S guidelines, our clinical strategy and our preclinical strategy should be in line with that. When it is in line with your ICH, so it may be applicable to throughout the world. So we can adapt the ICH guidelines. Accordingly, we can go preparation of our protocol, preparation of the or planning brochure, and conducting of your preclinical trials and clinical trials. So we should generate the same reports. At the same time, when the respect to country of your filing for the EU region or maybe FDA region, our protocol going to approve by them only. So that whatever you mentioned the protocol, if there is any gap, it is not in line with the guideline, they will propose this is required. So then we have to include it. Then before conducting study itself in the protocol, we are in line with their requirement. Then we have to fulfill that requirement, the continuing by studies. Got it?
Actually, here I want to understand here. For example, EMA required 500 patient data for clinical report.
Right.
If I conduct 50% from India and 50% from Europe, the data will be acceptable, or like all from
Acceptable,
Europe only?
acceptable. When you're going to conduct like global or maybe in the EU region specific or global clinical trial, 2 ways will be there, either country specific. When it is mentioned country specific, we should stick with the only country specific, that respect to country region people, or region specific with that regions. Where there is a global clinical trial, we should choose worldwide. So some people here in Asia countries, some people from Africa countries, some people from US, some people from Europe. Like there will be population should be selected partially. Then cumulatively, where it is mentioned 500 people, 300 people, or maybe can conclude from any extrapolation also can be included. Yeah, that we'll discuss during our, it is too early to discuss that point. When you're going for non-clinical overview and clinical overview, there will explain detail. Yeah, we'll have a separate session for that.
Okay.
Right?
Thank you.
Yeah, yeah. So now with respect to this model, even though on demo class also I explained, but even though it is required to know why it is we are going to discuss for the 40 sessions and technical sessions. So if you see model 1 is will come later, but we should know that this is related to patents, related to cover letters, related to when you're going for the file, any for the, you are going for the preclinical application, with you have to keep it in the data in the model 1. When you're going for the clinical application, we have to keep the product, the respect to documentation activity should be keep here, which is the non-technical part. That is what these are not part of clinical technical common technical documents. Model 2 to model 5 is called as a common technical documents, where we are going to discuss about, we are going to provide a data of technical information. So just forgot about model 2. We'll come back to the model 2 back. So in model 3 is a quality information. So when you go for quality information, there is a guideline is called M4Q. That means multidisciplinary quality guidelines. In that quality guidelines, to prepare the dossier, what information is required, what data to be placed, very clearly mentioned in the section wise. So to update to, or maybe to prepare that section wise, we should require a technical data. It may be data from R&D, data from QC, data from manufacturing, or data from QA. So we have to collect it to prepare this dossier section. So coming 40 sessions, we will be discussing on this same thing. So some data may be relevant to this, some data may be relevant to this. And after that, here we are going to part of drug substance, part of drug product, even though engineering part also something need to be keep it here, because the building infrastructures like appendices, equipments, and building infrastructures and clean room area classifications also be part of it. And biosimilarity also with respect to regional information also part of it. There will be 4 parts. One will be drug substance, one is a drug product and appendices, and there is a respect to regional information, 3 to R sections. So that the entire information we should be part of our manufacturing source only. Entire part will be manufacturing source. So from R&D to QC to manufacturing, entire quality assurance department going to provide the data. We'll collect it as a regulatory department and we'll refer the guidelines accordingly. Those will be prepared. So that we're going to learn from coming days. Now non-clinical thing with respect to animal studies, and the reports protocol will be get approved, and we'll hire any CROs, and CRO will be conduct the studies and we'll get the reports. The reports will be reviewed by themselves. At the same time, every organization will having a clinical team. The clinical team will review this complete part of non-clinical study reports, and they will be accepted, and the reports will be given to the regulatory persons. And regulatory team, so who is preparing the dossiers and who is going to submission, those people ensure as per M4, the safety guidelines. According to that, the sections has been developed and enclosed. So only the reports going to enclose here with respect to which is already someone is approved by clinical team. Similarly, clinical part also, the complete as per M4S guidelines. I will show that guideline end of the session. So we will refer that guidelines and we'll go for accordingly those development and will be enclosed here. So now model 4, model 5 will be with respect to very minimal. The people working in the model 3 very rarely will be open this one. Most of the people of quality dosage preparation, people CMC model 3 preparation will not be part of it. Maybe there is a divisions in the regulatory affairs department. So some people will be preparing the quality dossiers, and some people will be part of submissions of your regulatory market submissions. Some people will be taking care of domestic markets. Some people will be taking care of, some other team will be taking care of collection with the coordinate with the clinical team, collect the dossier. So like this, different teams will be there in the regulatory affairs team. And when you're going to prepare model 3, model 4, model 5 is ready, is a part of R&D development, part of commercial manufacturing, part of clinical manufacturing. 3 stages of product development will be placed here. And part of non-clinical part, preclinical studies going to place here, and clinical part phase 1, phase 2 if applicable, then phase 3 data will be parking here. Then against that, we are going to open the model 2 and we are going to provide a quality overall summary. Here maybe 2000, 3000 pages of documents and having around 64 sections, and each section having, one of the section having 1 page, one of the section having 100 pages. So if you cumulatively, if you take total 64 sections of model 3 documents, it may form 3000 or maybe 2000 to 3000 pages of documents. When you dump this document for reviewer to review the process, it may be difficult for them. For that reason, you are going to provide a overview of this model 3. In that overview, a single document containing a limit of pages, 40 to 80 pages. In that one, clearly going to write up how many batches, what is the batch scale definition, what is the, how many batches taken at the preclinical stage, how many batches are taken in the clinical stage, how many batches taken in the process validation batches, and what is the stability shelf life, what is the current stability available time points, how many batch lots for the biosimilarity, part of analytical similarity has been conducted. Everything will be written here with respect to DS, with respect to DP stability point of view and batch scale definitions. So the reviewer want to refer the stability. He will straightforward open the respect to drug substance section of stability with respect to section of the drug product stability section, and he will review it. So that is a one type of index. So where we can easily can we see where is the information available in this section. So accordingly, the review process will be happen when you submit the dosage. Similarly, non-clinical also having thousands of pages of documents. So there is a non-clinical overview, non-clinical summary is there very clearly. So instead of reviewing this document straightforward, the reviewers will be review first this one, understand the overview, understand the summary, then straightforward they'll be open here. Similarly for clinical also, the clinical submitted those years, what is the reports are there. They will check the review process overview on the clinical documents as well as clinical study reports. Then they will be go for respect to those sections. Like this, model 1, model 2, model 3, model 4, model 5 is the aligned as per ICH guidelines. So as per ICH guidelines. So most probably the prepared dossier as per ICH guidelines, what are the prepared here? Once you prepare the technical document, so this is enough to submit worldwide. So there is a separate country specific guidelines are there. They want to their format. But what you put in the technical information here in the model 3, model 4, model 5, there is no changes. With respect to changes, maybe numbering system or maybe some terminologies, but no changes in the not if the product is approved in the EMA and you are looking for the Asia countries, let's say Malaysia or maybe Singapore. So the Singapore or Asia or maybe Malaysia country is going to accept to submit in the CDD. Need not worry. The prepared dossier which is for the EMA, we can submit for the Malaysia as well. But in case of as per Asian guidelines, they want to in the ACD format. So there is a terminology changes and certain modification of the numbering system. That is also part of our sessions. So CD also can learn, ACD also can be learned. Now moving forward to the next slide. So which is CDD structure. As we discussed in the previous slide, there is the CDD structure numbering system, which is given as a model 1 as a start with model 1. There is a model 3 start with 3. 3 stand for model 3 and 1 stand for table of contents. And after that, the numbering system will be 3.2, 3.2.1 drug substances, 3.2.S, 3.2.P drug product. Like that, there is a numbering system. Initially, it may looks like difficult to understand that numbering system. After spending of time for 3 months, you can easily understand the numbering system. It will be easy to catch it. Similarly, 4 stand for module 4, 5 stand for module 5. So this is what we're going to learn, how to write the dosages in the CTD format and which data where you have to going to keep it. Now the question is, how it is ICD structure? If you see ICD structure, there are 10 countries in the Asian region, so including Malaysia and Singapore, and there is Indonesia, and there are 10 countries are there where we're having a separate session. There we can see individual how many countries are out of 10 countries who are going to accept it. Instead of 5 modules, they're having a 4 parts. So there is a 1 module was less here. But the part 2 is nothing but your module 3. And part 3 nothing but your module 4. And part 4 nothing but module 5 only. Here, the quality overall summary is including of your part 2. And non-clinical summary is a part of your initial of the documentation, initial of the dossier is a part of 3. And your clinical summary part 4 is a part of it, is measured here. So to reduce the documentation activity, because same information what you given here of 3000 documents, here may be going to provide in the 40-50 pages of document or maybe 80 pages of the document. So they removed, as per ACD guidelines, they removed this one and they included it in the part of module 3 only, in the part of your part 2 as a quality document. So there is no need to refer other documents. The reviewer which is going for the review for the module 3 dossier, so he will refer, especially with the part 2 quality part, he will see or she will see first table of content and summary, then go for the body of data. So the reference summary will be available here. Similarly, part 3, part 4 also, non-clinical summaries and non-clinical clinical part will be available here. So there is only 4 parts. And how you submitting module 1 with respect to non-CTD part, which is a technical, non-technical documents, administrative documents. Similarly, applicable for part 1, administrative data, which is called as a non-technical documents. So there is not much differentiation. So as of now, just keep it in mind. So last at the going forward, we having a separate session for it. The prepared CTD dossier, how we are going to convert the ACD dossier. The people already in the regulatory field, they may be aware of it. It was very easy. Nilesh, are you preparing any ACTD dossiers?
We had submitted a lot of them previously to APEC region.
Yeah, once you prepare CTD, is it any difficult to prepare ACTD?
No, it is very easy. In fact, I mean, it is just change of some headers and footers, and I think we can manage that.
Correct.
Pretty much easy.
Yeah. So no need to worry. So we'll going forward, we'll learn it in the coming sessions. Yeah.
Okay. I want to ask. So if India is submitting a dossier to Ghana, Ghana, we look at the CTD with the module 2 format. Would India submit it in an ACTD format or to the country specific format?
Yeah, in Africa countries, there is a guidelines, right? They want to, respectively module 2, in Africa countries, there is a separate guidelines. So what we are providing data by referring, if you prepare in the CTD format, when you open the country specific, that entire information available here only. We have to transcribe that information as per the checklist. That is also very easy. Need not to worry. The Africa country, they will ask as per their MOH regions respect to guidelines. Maybe may not be changed in the module 3, module 4, module 5. With respect to module 2, their own format will be there.
Maybe, just to add, sir, like maybe the technically, the contents will be remaining the same. However, the arrangement of those contents in the respective dossier sections, as you said, will be in the country specific formats if there are any in particular.
Correct. Right.
Yeah.
Clear. Yeah. Hope everyone is clear so far.
Yeah, it's okay.
Yeah. What about others? Everyone is following with me?
Yeah.
Sorry?
Everyone is following with me?
Yeah, we are following.
Yeah, fine. Yeah. Now, if you see here, different molecules I place it for just understanding. So as we mentioned in the respect to syllabus, we are going to discuss about 3 type of products. Virendra, can you meet? Yeah. So in our product portfolio, with respect to our training session, we're going to take up 3 products. In that, one is the insulins, another one is the monoclonal antibody, and vaccines. So vaccines also we're going to take up like conjugated vaccines. So that these are the 3 biological leading molecules. If you see in the everywhere, there is a part of immunization process, vaccines are mandatory. So that after learning of this course, the scope for the industry entry or maybe switching to regulatory. So there is a very easy to enter into the vaccine industry. So how we are manufacturing vaccines, we can how to file the vaccine production in the respective MOH, we can clearly understand. At the same time, today the leading molecule was monoclonal antibodies. So especially for the oncotherapies. So once upon a time, these monoclonal antibodies was very costly medicines, and 20 years back, if you want to go for any oncotherapy, it's not available in the every country. It was dedicatedly available in the maybe US region or maybe EMA somewhere. So the people has to travel there and take treatment and come back to their native place. Nowadays, after 20 years, the scenario was entire thing is changed. Now, the biosimilars of these oncological products, the innovator product was replaced by biosimilars. So just imagine one vial is a 1 lakh rupee of Indian rupee. The biosimilar product is available at 20,000 rupees of vial. So that the government also can support and the patient also can affordable that price, so that the people who suffering with any oncological issues, so they can go for therapies and they can get cured. So similarly, in case of insulin products, so the insulin analogs, we have 4 to 6 type of analogs, which is required for the part of diabetic blood sugar level control. So in that case, earlier it was there is a insulin human, but it's not in case of any biosimilar product as of now. So insulin glargine, so which is the modified version of human insulin. So that product is reported, that product registered as a Lantus brand name in the US as a Lantus brand. Against that, nowadays we have got it. Many products are there in the as a biosimilar products, being in the FDA registered products also, like Semicli with Biocon brand name. So that is also available here. So now why want to discuss about insulin products. So after vaccines, and the most demanding molecule was insulin. There was worldwide many patients are there suffering with diabetic. So there are many manufacturing companies with respect to drug substance, with respect to drug product preparations, and there is a with respect to any part of any CMO or CROs are there working on the insulin related organizations, the manufacturing related this product. So that this will give us also a lot of job opportunities as a biological products, biosimilar products, insulin background products. So these sessions can help how to construct related to your microbial process and related to cell culture products and any viral related products in the this session can be teach us so how to prepare the cell banking system, how to do upstream process, how to do manufacturing process, and how to file the regulatory pathways. Now, with respect to small molecules, this is wherever is required. Wherever is required, we will refer the small molecule also to comparison purpose. There is a question from some people: is it possible to teach a small molecule also? Yes, definitely. But when you're going to learn biological process, this is very easy, pretty easy. So now the differentiation between biological product versus the small molecules. If you see the size of the molecule is them less than 1 Dalton, or we can call it 1,000 Daltons, or maybe 1 kDa. In case of large molecules, the size which is the biological product is a more than 10 or maybe above 1 kDa also can be concluded. Some peptides or anything can be fall under biological products. The source of these small molecules is a chemical synthesis. That means the chemical which you are going to do some hydrolysis or do some chemical reactions, we can able to produce some substance to product. But in case of biological products, it is a cultures of living cells. So we are going to use any Chinese hamster ovary cells like CHO cells, any HEK cells, human embryonic kidney cells, or maybe some microbial living cells, or maybe bacterial cells, whatever maybe. We are going to produce a biological product from a living cells, which will be safe and no harmful for the human body. In case of chemical synthetic products, it may be affecting, but having any adverse events, it may not be at the moment. Later stage of our body going to expose any adverse events, or maybe side effects can able to see in the later stages. So that is the reason now the small molecules are replacing with your biological products. Small molecules are replacing with biological products, and it will be generally orals and solids in case of small molecules. In case of biological products, most of the products are injected or infused. So injected may be subcutaneous or maybe intradermal or intramuscular. In case of infused, it's intravenous injections. So especially for the MAP therapies, required to treat 4 hours, 6 hours continuously circulating through infused solutions. So it is called as a infused. It may be dispensed by the pharmacist. If you go and ask by oral also, they will give the this dispensing medicines can be given. If you go and ask paracetamol, they can easily dispense. If you go and ask any antibiotic, there is a possibility to give it without any prescription. But in case of these biological products, without doctor prescription, the medical dispenser, the medical pharmacist is not going to dispense this medicine. Like examples, we can see many chemical products, many chemical small molecules, which is a parental, or we can call it as a small molecules, chemically synthesized products. We can able to see tablet forms, ointments, any solutions kind of thing is part of our protein requirement. In case of biological products, one of the example is given a monoclonal antibody, which is Herceptin, the brand name, which is a using for Herceptin as a breast cancer treatment. So nothing but it is a trastuzumab, which is a trastuzumab. Nowadays, most of the female bodies is impacting with suffering with respect to breast cancer therapies. So required this type of monoclonal antibody, treat them and protect them from this kind of onco-related issues. So like this, going forward, we are going to discuss one of the product is insulin, another product of monoclonal antibodies, and another product is a vaccines. So one is a cell culture product, one will take a microbial product, another one is a viral based vaccines, or many microbial vaccines will be taken up. So these three products will give a lot of knowledge for the related upstream, related downstream, anti-drug substance, drug product, and preparation of the analytical part, and preparation of dossiers, and sub-filing of the part of your product. So these three products will give complete overview, so that if you understand these three products, you can deal with any type of product. It may be we are learning insulins, learning monoclonal antibodies, and maybe learning about vaccines. Suddenly we are going for peptide filing activities, peptide products also can be done. We are going for small molecules, pretty easy. If you understand these large molecules, the small molecule process very easy. So going forward, we're going to learn these products. Yeah. Now I explained this slide previously also, part of our demonstration, but still I want to give it some knowledge about the insulin uptake mechanism, how link with our regarding biosimilar pathways, we are going to discuss. So here, so when you take it a mixed meal, it may having carbohydrates or maybe proteins. So the proteins and carbohydrates will be going to enter into digestion system and going to produce, after digestion, going to produce glucose. So there is a lot of biological mechanism going to produce a glucose. When that glucose want to take uptake by our body tissues for the part of biological process, so required insulin receptor mechanism. When insulin is going to recognize this, here the receptor of the cell body on the cell surface, then the port will be open where glucose can be transported. So when the where the insulin is going to produce, the insulin is going to produce in the pancreas, which is having a beta isocyte cells. So that will be a support for the synthesis of insulin. The produced insulin go and circulate. It will be stored in the liver, as well as it will go in the surface of the going to react in the surface area of the cell, particular body cell. Then there will be port will be open. So automatically glucose, the produced glucose will be entering the cell, inside the cell body. Then we will start the mechanism. So blood sugar regulations. After you eat, your blood sugar levels will be raised generally. So the insulin is released to the help to lower these levels by moving glucose into cells. So once it is moving to the cells, energy storage can happen. If these excess glucose, insulin helps to store in the liver and muscles, form the glycogen. If the glycogen stores are fully, it converts into glucose into fat or for the long-term storage. So it is a balanced metabolism. Insulin ensures that body has been steady supply energy by regulating how glucose is used and stored. So it is happening in the natural mechanism of the body. In case this pancreas beta isolate cells, part of genetical issues or part of aging effect of the body, it is failed. It may be partially failed or maybe completely failed to synthesize insulin. When it is a partially failed or maybe fully failed of synthesizing of insulin, when the body is taking a meal, automatic glucose is producing. The glucose is not going to take up inside of the cell. Automatically, the glucose level in the bloodstream is going to increase. So that is automatically called as a sugar levels is increased. When sugar levels are increased, imbalance and patient, the body will go several diseases, uncontrolled and unable to walk, unable to stand, unable to, brain can't work properly. So many unwanted diseases will come. Now, to avoid that, the patient anyhow, due to aging effect or maybe due to genetical issue disorders, so the patient impacted with the defect of insulin synthesis. Now, what will the scientists develop? The biological products through biological systems, they produce a insulin injection. So the insulin injection, when I inject into the body, in the subcutaneous, when it inject into the body, the insulin which is containing the solution, acidic solution in the unit basis. When it is injected into the body of low acidic solution, our body pH will be around 6 to 7, and the solution having around 4 to 4 around 4. When it is injected here, that will form as a microprecipitates, because some crystallization will happen. Microprecipitates. The microprecipitates will travel in the bloodstream and will be reaching into the respective receptor, and automatically glucose transportation will be taken place. So that is the reason the patients who are suffering with any insulin deficiency, the before meal, they will get injection. Is it correct? Hope everyone is following with me.
Yeah. Very clear.
Yeah. So like this, we should be understand the molecule which we are discussing about insulin, with the molecule which we are discussing about vaccine, the molecule which we are going to discuss about MAPS. We should understand the mode of mechanism. This is called a mode of mechanism, because part of our dosage development, part of our understanding, part of our development process, we should understand, so that the insulins are, there are many patients in the worldwide related to the deficiency with our insulin deficiency. There was many patients with diabetic reported. So that if you develop this kind of insulin molecules, there is a market. So organization strategy should be think about that, should be develop this kind of products, so that we can able to market. We can develop, we can go for market, and organization can be grow automatically. Organization can be give up the job source for the employees. So this is for just for understanding. Now what is biologic? So can someone can give definition of the biologic? So it is a interactive session. Please interact and be inactive and learn a lot. Then only we can learn. So one side will be go. Then you have to raise questions and answers can be easily can get it. Yeah, someone give definition for biologic. Sorry.
Biologics are something like
Yes, madam.
Biologics are something like that will be synthesized in the body.
Correct.
When it comes to the biosimilars, in vitro conditions, the same molecule will be synthesized as in the form of as it is formed in the in vivo conditions.
Okay, somewhat relevant. Yes, someone want to add up?
Actually, biologics are the drugs, the substances which are name itself indicates the biologics. These are the made up of bio means these are the products synthesized or prepared with the help of the bio proteins or monoclonal antibodies or any other type of like microbes or through cultures. So these are like biologics. Means name itself indicates these are the biologics. So the main source of these drugs, like just like antibiotics, like the main source of these drugs are biologics. Source.
Yeah, fine.
Biosource.
Yeah.
Biosource.
Yeah.
We can
So
say.
the medicinal product where you are going to produce by using living things is called as a biologics. So the product which is registered and approved in the respective regulated markets is going to call as a either innovative product or can call as a biologic. The biologic is when you're going to produce a biosimilar, then the biologic molecule is called as a reference biologic. So what definition is saying that the biological products are regulated by Food and Drug Administration (FDA) and are used to diagnose or prevent, treat, cure diseases, medicinal conditions. Biological products. As the example of a given insulin glargine, which is approved as a biological product, against that we produce a biosimilar product. So biological products are diverse category of products and are generally large, as I shown the size of the variations. If you see chemical drug, which are aspirin, around 180 Dalton, whereas insulin molecule around 5,800 Daltons. Whereas you are talking about monoclonal antibody around 150,000 Daltons. So the complexity of the molecule is a large. That is what he's saying. Generally large and complex molecules. It is a combination of many amino acids, many groups of amino acids. And these products may be produced through biotechnology by living systems. So some of the products may be required to modify the organism genetic system. There are some products without modification also, naturally available strain, which can be produced. Some natural strains can be produced. Some medicinal products as a secondary metabolites. So we can isolate those kind of living things and we can go for we can go for industrial application of commercial manufacturing. Those also part of vaccine. We're going to discuss that. Those generally we call as a genetically non-genetically modified organisms, which you are not going to disturb their genetic system. Even though we are going to call as a biological products, such as microorganisms, plant cells, animal cells, and are often more difficult to characterize than small molecule drugs. We can't easy to characterize that much easy as like small molecules. There are many types of biological products approved in the US, in the United States, including therapeutic products such as filgrastim, monoclonal antibodies such as adalimumab, trastuzumab, bevacizumab, rituximab. There are many maps out there in that cases. And infliximab, which is our case study going to discuss, infliximab. And vaccines such as those are for the influenza, tetanus. There are vaccines as a biological products. Now, the definition what is saying that what is meant by reference product. So now biological product is reached in the FDA region or maybe EMA region. So the reference product, what is we are going to call it? Why we have to call it a reference product? When you want to make a biosimilar product, we are going to refer this product for the biosimilarity assessment, part of analytical similarity, part of clinical similarity, part of preclinical similarity. So we are taking as a reference. It is a standard to make this product and going to ultimately finally the product which you are making here, that should be equal to this. That is the reason this product is termed as a reference product. Definition what is saying? A reference product is the single biological product already approved by FDA against which proposed biosimilar product is compared. Which biosimilar product is compared. That means this is already reached in the FDA. Against that we produce a biosimilar. For the reference of this one, we used as a biological product, and the terminology was used as a reference product. Now, there is a biosimilar. What is the biosimilar product? A biosimilar is a biological product. This is also how we are making this product by using a living things. This product also going to produce similar way. We can use different platforms. This product may be made E. coli based. This product may be made yeast based. This product may be made in the like mammalian cell culture, HEK cells. This product can be made by using CHO cells. There is no necessary how the innovator product is made. Similarly, you have to use the same platform. We can use the manufacturing ability how you want, how you can develop, that is manufacturing choice. But ultimately, the final quality attributes, which is we made a QTPP document, quality target profile document, that should be complies this versus this. Then only it can be claimed as a biosimilar product, comparable, highly similar conditions. So what definition is saying? A biosimilar is a biological product that is highly similar and has no clinical meaningful. So difference in the from existing FDA approved product and the reference product. So when you're going to conduct quality comparison and we're going to conduct preclinical and clinical, there should not be no clinical meaningful. That means this product should not show any variation about this. So the first we'll say quality part, nothing but analytical similarity. We'll ensure before going to preclinical, we'll ensure analytical similarity. That is what there is a R&D batches which you made prior to going to preclinical protocol. Will produce 3+3 data. In this 3+3, 3 lots of manufacturers, 3 lots of our reference biologic. We'll use for the quality comparison, and we'll say the quality comparison is inline. Inline means when it's saying this product is 98% pure, this also 98% pure. This may be 99, this may be 98.5 also can acceptable. It should be similar, not the not out of specification of our reference biologic. It should be within the acceptance criteria. Maybe slightly variation, it may be 99%, it may be 98.8%, or maybe 99.2 also. It can be better, it can be close to similar. So that way, this part was made and ensured analytically similar. When analytically similar, we can expect preclinical also similar. The adverse events may not be going to happen. When analytically is not similar, there is a percentage of aggregates is more in the biosimilar product. Percentage of aggregates is more. It may be show immunogenicity reaction, so that the non-clinical part may be. So we can't estimate that will be safe for the preclinical point of view. So agency will review first analytical similarity. When it is a percentage of aggregates is mentioned not more than 2%, and this product is 1%. This product may be 1.2 or maybe 0.8 or maybe 0.5 or maybe 1.5 also can be acceptable. It is a similar. So there should not be any clinically meaningful. This is called as a biosimilar product. Is it clear now? To reference product means definition and biosimilar product means definition.
Yeah, yes, but what about if at the end of the day the biosimilar is having better efficacy, or like is having better therapeutics than the main or the reference product, do you still consider it as a biosimilar or a bio-better?
Bio-better.
But it is still a biosimilar, even though it has everything the same.
Still we can claim as a biosimilar product only because the product is already listed, but it will be claimed as a bio-better, marked as a bio-better. So the usage of the product, the demand of the product will be more. How we can go, we can claim it. So we have to prove it. Preclinically should show better version, and clinical also should be better version, and phase 4 data also should show better version. That means efficacy rate should be increased. That usage of population should be increased. The preference or the choice of this product by physician should be increased. Then we can claim. It will take time. Immediately we can't claim, even though clinically showing better and preclinical also showing better, analytical similarity also showing better. But based on the phase 4 data from real data, we have to prove it. So after some time, we can get it called as a bio-better molecule. When it is reaching to the patient, then collection of phase 4 data we can claim as a bio-better.
Okay.
Yeah.
Okay. Yeah.
Yeah.
Clear.
Sir, I have a question, sir.
Yeah, please.
So somewhere we see there's a written reference product, and somewhere it is written RLD, reference listed drug. Is there any difference?
Nothing difference. Either you can call reference product, innovator product, reference medicinal product, or we can use biological product. There is many terms there. Ultimately, the term is a one thing only. Ultimately, that is a one product. RMP, RLD, and you can call it as a biological product, innovator product. All are same only. Clear? Javier, is it Javier?
Yeah.
Okay. Are you in
Yeah,
WhatsApp group, Javier?
Yeah, fortunate. So, sorry to interrupt. I want to be clear. Yeah, I want to be clear. Biosimilar and biologics, I mean, the comparison, is it to, how do you call it, drug substance or to drug product, or both of them? A drug substance can also have a reference drug substance and a biosimilar drug substance, or a drug product can have a reference drug product or and then a biosimilar drug product, or it can go for any of them.
Yeah, very good question. See, when you're going to perform biosimilar assessment, we should use drug product versus drug product similar assessment. So when you want to protein characterization, drug substance versus drug substance has to be performed. So the drug substance from innovator product is not able to get it. In that case, what we'll do, we'll procure drug product and we'll do reverse engineering. Reverse engineering means we will remove all the excipients and we'll make it as equal to your reference product as equal to your drug substance. Then we'll go for quality comparison, protein characterization point of view. That is also part of our sessions. We'll cover in going forward in detail.
Okay, thank you.
Yeah.
Can I ask? Can I interfere?
Yeah. Yes, Virendra.
Yeah, regarding, as you told, for example, for biologics versus biosimilar. So as you told, second type, the kind of sources, it may be yeast or something. So what price difference with this? What is the cost impact? And can you give one example?
That will be no. Either you produce E. coli or produce yeast. There is no any cost effective things. So the price of the cost will be remain same. What is the government is fixed against biological product, against your innovator product, that has to be followed. That has to be taken care by manufacturers. Yeah, if you produce yeast means the cost will be less. If produce means E. coli means cost will be high, means not like that. The manufacturer is going to choose the way of manufacturing operation is easy for him. Accordingly, we will choose which maybe in yeast process getting high yield, or maybe in E. coli getting high yield, or maybe scale up point of view, yeast is supporting for large scale. E. coli is not supporting for large scale supportation. According to that, you will choose the platform.
Can you give one latest example with some biological name and biosimilar? So how the complication
like there is a given example like biologic is a Lantus, which is the innovator brand insulin glargine product.
Okay. Lantus.
And again that now today Biocon is produced, which is called as a biosimilar product, which is a Semigly brand, which is approved by FDA. So that is called as a biosimilar. Now, is it clear, Virendra? You stay back. Yeah, you stay back. All the points I'll clarify in the session. Okay.
Fine.
Yeah. Now the third thing is interchangeable biosimilar. Do you know anyone what is meant by interchangeable biosimilar?
So wherein the physicians can certainly certify that, like they can change their existing reference product therapy by the available new biosimilar product. They can trust this, and the authority also kind of provide that designation to the product which is a biosimilar product as such.
Okay, fine. Yeah, I will add.
Sir, regarding interchangeable, any extra addition of treatment, as you told, I want to give example with respect to allopathic example. The flurifloxacin is their example. It is used for medication of urinary tract infection. When you extending its treatment for traveler disease or something, that is one kind of extra additional requirement apart from therapeutic primary indication.
Okay, fine. No worry. I will decode it.
Is it I'm okay? What I told example.
Okay, it's fine. I will decode.
I've given example to allopathic medicine. So
Okay.
how this is the additional requirement is add-on existing the primary therapeutic requirement.
Agreed. Yeah. So coming to biological things, so we are talking about interchangeable biosimilar. An interchangeable product, an interchangeable biosimilar is a product like biosimilar. It is like a nothing but biosimilar only, but meets additional requirements. The additional requirements during our clinical phase. Yeah, please. Yeah. So respect to interchangeable product, when we require to conduct additional studies, substitutional studies during our clinical studies. So when it is approved as an interchangeable biosimilar, when it is approved as an interchangeable biosimilar, so it will be replace this reference product. So when it is approved as a biosimilar product, the physician can be support to use this one, then only you can use. When physician is suggesting to use reference product, the patient should be use the reference product only. When your product is registered as a interchangeable biosimilar, so even though physician is given this product is to use the patient, the pharmacist doesn't have the stock of this one, but he having a this product. Instead of this, you can use this one. You can dispense this one. Case 1. Case 2, when you're having a pharmacist, the doctor is prescribed as a to use the reference product. But when entering into the pharmacy, the pharmacy is shown in place of in front of you. You can choose this product or this product. This product is having a $100 and this product having a $60. Which one you want? So the patient can be having an option. Either you can choose interchangeable biosimilar or you can go with reference product. And let's imagine this reference product lost since 10 years the patient was using, and suddenly you can switch over to interchangeable biosimilar. But coming to the biosimilars, there is a physician proposal is required to use it. That is what the difference of interchangeable biosimilar. Interchangeable product is a biosimilar product that meets additional requirements. The additional requirement is nothing but during clinical studies, we have to be conduct a substitutional studies. That is a additional requirement.
Excuse me.
Yeah, please.
I'm having a doubt here. Can we call this interchangeable product as a generic?
No, madam. These are all
that is required.
Yeah, required, because generic, when talking about generic, so there is no clinical studies, there is no preclinical studies and all. Once you've done bioequivalent studies, the product can be launched in the market. But in this case, required preclinical studies, required clinical studies. In case of interchangeable biosimilars, required additional substitution studies as well.
Okay, thank you.
Yeah.
Includes an additional substitution study. What the studies include?
Virender, that will discuss coming days. No worry. Yeah. So now we're entering into how we are going to make it this biological products. So since it is in a overview, so what are the insulin you want to produce? We have to isolate the genome. From that gene, we have to cut it. We have to go for vector system. We have to construct the vector. So tomorrow onwards, we're going to talk about cell banks. So there will go in detail of vector system constructions, because part of submissions, we require to know which type of genome you are taking and how you are going to purify the gene and how you are inserting in the vector system, how we are going to make a cell banking system. What is the requirement of two-tiering of cell banking system? How we are going to characterize it? Characterization, what is the criteria? Which guidelines is referred? Everything you want to refer it. Once cell bank is made, going for upstream studies, then downstream purifications will make a final purified bulk. Going forward, 40 sessions, we are going to discuss about cell banks for 5 sessions, upstream for 4 to 5 sessions, and downstream for 10 to 15 sessions. So we'll come to know that complete drug substance strategies, CMC strategies. So that is what coming 25 sessions, we'll going to discuss complete CMC. We'll come to know that how to produce and how to purify it, what is the purification strategies, and what type of strategies to be used to produce the CMC data. Now, so using a reference or biosimilar or interchangeable biosimilar products. So here the patient and healthcare provides can be confident in the safety, effectiveness of biosimilar or interchangeable product and prescribe them by name just for the reference product. So everything, either you are going to use biological product, you are going to use a biosimilar product, you are going to interchangeable product. The point of view, we have to see safety point of view, effectiveness point of view. So if this safety is not there or effectiveness is not there, even though purity is meeting, there is no matter, the product will be no valuable in the market. So the biosimilars and interchangeable biosimilars products can be used a patient who have previously been treated. So the previously, as I given example in the previous slide, the same statement is written here. And the reference product, so treatment will be experienced, and the patient who have not previously been treated with the reference product, we can use a interchangeable biosimilar. And interchangeable biosimilars may substitute for the reference product without their intervention of the prescribing healthcare provider, nothing but doctors, subject to state laws. Already FDA has been, the regulations has been provided here. So it should be required, meets FDA rigorous approval standards and safe options for the patients and effective options for the patients. This is the requirement of your interchangeable biosimilars. When it is meeting these three criteria, this product can be approved as a interchangeable biosimilar. Now, if you see, there are over 41 biosimilars or more than that also available in the market, approved by FDA. From that, if you see, these are the B stand for biosimilar. B stand for biosimilar. In this case, supportive care, oncology care, and related to autoimmune system, and there is a ophthalmology products. From that, there is I stand for interchangeable biosimilar. There are only 3 products from that insulin glargine having 2 products, 2 companies. From that, Biocon is the one of the company is get approval for the interchangeable biosimilar as a insulin glargine. So out of this 41 biosimilars, we are having a 4 products as a 4 companies is going to make a interchangeable biosimilars. From that, only 3 products is there. In that, one of the Indian manufacturing company is approved in the FDA region. Yeah. Now, why strictly interchangeables is required? So interchangeable biosimilar product can be substituted for the reference product. Straightforward, we can remove the innovator product and we can start usage of this product. The pharmacist without intervention of the prescribing healthcare provider, subject to state pharmacy law. So when the doctor is prescribed as a to use the reference product, when it is reaching, the script goes to the pharmacy, and the pharmacist substitutes the interchangeable biosimilars for the reference product. We can do it. He is having a option to give it. Patient can receive the interchangeable biosimilar and there is no any doubt on it. But in case of biosimilars, doctor has to prescribe it and pharmacy person has to be dispensed the same. Then only it will be given to the patient and patient can be go forward. So if you prepare the product cost wise, the biological product having a $100 and interchangeable biosimilar may having $60 and biosimilar product may having $20. Now the insulin glargine of Semiglear brand as approved in the FDA region, which is Semiglear brand of Biocon product, which is the biosimilar product, is having around 20% cost of the innovator product. And this interchangeable biosimilar having around 60-70% cost of the innovator product. So that's why the manufacturers will invest money to generate the clinical data additionally and get additional point of benefit regarding revenue generation. Now, the high product quality stands. Biosimilars is having biosimilars analytical similarity as well as non-clinical meaningful differences is required for the biosimilars. In case of interchangeable biosimilars, there is a statutory criteria to related to the potential substitutional without intervention of the prescriber, any given patient, risk of altering the any switching studies. These are all when you provide additional data, this biosimilar product can be reached as a interchangeable biosimilar. Going to extra studies is required in the part of clinical studies. So the biosimilars and interchangeable biosimilars, the applicants request licenses as per biosimilars and interchangeable biosimilars. An applicant may be first approved as a biosimilar, but later can seek interchangeable biosimilarity. So most of the manufacturers, they will try to register as a biosimilars and later they will keep on study and continue to claim as a interchangeable biosimilar. So maybe this product is registered in the biosimilar and selling at $20 and later after providing as traditional studies of your interchangeable biosimilar studies and going to get it extra data, then we can go for register as a interchangeable biosimilar which is selling at $20, this cost will be converted into $60, where 3 times is higher. So one time of study of investment regarding your clinical study can triple the organization revenue. So that point of view, organization get good name, and the product can be survive in it can be save the patient's life, available at cheaper price than innovator product. So that point of view, the sponsor organization going to help for the patient life. Yeah. The analytical similarity and product quality standards are the same for the biosimilars and interchangeable biosimilars. There is nothing to difference. Only the thing is additional clinical studies has to be conducted to substitutional studies. That is what the requirement of interchangeable biosimilars. Now, the statutory criteria are related to the potential for substitution without intervention of the prescriber. This is the benefit. So when you're having a interchangeable biosimilar and innovator product and biosimilar, the pharmacist and the patient can choose which one he want to or she want to take it. That is the benefit of interchangeable biosimilars. Yeah. So now to develop either it's a biological product where you want to develop.
I'm having a question.
Yes, please, madam.
Please go to the previous slide. Okay, thank you. And when you say that for interchangeable biosimilars, additional tests will be carried out, right?
All thing is required, madam, to conduct. It is required to analytical similarity to be provided, and clinical and preclinical also required. And we have to show there is no clinical meaningful differences with against your reference product, and we have to claim as a biosimilar.
Yes, yes. For interchangeable biosimilars, what are the additional? Yes, what are the additional tests we can add in this?
Yeah, substitutional studies. Part of our clinical, we will come to know. Substitutional studies, additional clinical studies is required. As per biosimilar guidelines, this is enough. When you want to claim as interchangeable biosimilar, we have to refer additional clinical studies. From that, we have to prove the evidences. When you prove the evidences, we can add up the existing molecule. Already this product is registered in the FDA region. After conducting the studies, we will show to the data to file to this product as interchangeable biosimilar. FDA or maybe the FDA or maybe MOH will be review of this status and can be approved as a interchangeable biosimilar.
Okay, are there clinical studies which are going to do as an additional test? Is it depends on the product? I mean, product for which we are going for clinical trials, or any standard test will be there as a part of this interchangeable biosimilars?
Additional extra studies is required, madam. Part of additional substitution is a part of clinical.
Yes, yes. Additional study means it depends on the product or
any product. If you say take example of insulins, so using for the study is a part of diabetic treatment. If you take a MABs, going to treat for the part of our oncological treatment, any cancer therapies. So there in the phase 3, real patients we are going to take it. The people already suffering with our respective diseases. There we are going to substitute the comparison. We're going to use this our biosimilar product and going to calculate the efficacy, recovery, everything point of view equal to or more than that innovator product. So that point of view going to conduct additional substitution studies, and we can get it come as a registered as a interchangeable biosimilar.
I think we can discuss it later because
sure, madam.
Definitely. Thanks for
no, we can discuss later also. Yeah.
Yes,
that will come now part of our development, part of our again and again it will come. Yeah. Yeah. Now, as I show on the guidelines, M4Q guidelines with respect to Module 3 CMC data generation and M4ES guideline for Module 4 related to preclinical studies development, and regarding Module 4 M4E, which is guidelines for the our Module 5 data generation with respect to clinical trial requirement, and regarding refer ICH M4 guidelines, which is a multidisciplinary guidelines for CDD module organization. How we are going to organize those here. So today, too early to discuss this guideline, but we require to discuss this guideline another 5 minutes. Then we will go for Q and R. Is it okay to everybody?
Okay.
Yeah. Yeah. Yeah, the guideline is visible to everybody.
Yes.
Yeah. So whatever I mentioned their guidelines, there are 4 guidelines is mentioned as per ICH. So here if you see the common technical documents for the registration of our medicinal products, nothing but the common technical documents for human use research, pharmaceuticals for human use. So what data is required? Who should refer from R&D to till market? What guidelines is saying? Accordingly, we should reference ICS guidelines. Accordingly, we should develop the technical evidence documents for the part of your Model 3 quality preparation. So if you see here in that detail of reference guideline, so there are Model 2 and Model 3. Model 3 is the quality data. Model 2 is the your summaries, quality overall summary. Straightforward, we'll go to the Model 3. Then we'll understand. During our demonstration also shown partially this guideline, but I will show that how the technical sessions which we are going to discuss, how it is linked with here. So if you see Model 3, so what is the exact requirement of Model 3 requirement? So this document intended to provide guidance on the format of your registration application. So for the drug substance, their corresponding drug products as well as defined in the scope of the as per ICS guidelines Q6CA for new chemical entity for the chemical drugs. For the specification should be referred small molecules. In case of biological drugs, ICS guidelines Q6B for the biotech to be referred. So this M4Q guideline will give guidance for the development for the both the products. So with respect to our requirement, biologics should be referred as a only biotech products. So if you see, it is having 4 parts. From that, one is a Model 3 stand for Model 3. One is a table of content, TOC will be considered generally. From that body of data. From that body of data having 4 parts. So one part is a drug substance, another part is a drug product, where 3 stand for Model 3, 2 stand for body of data, 10 stand for drug substance. Under the drug substance, there are 7 sections, main sections. From that 7 sections, so each some are having single section, some having multiple subsections. So part of your general information. Under that general information, it will very simple. Even internet also it can be available. So what is the compoundal name of the relevant product? What is the chemical name? What is the company laboratory code? If you see in our organization, there will be coding system. So like product A, B, C, D like that, or maybe product 1, 2, 3, 4 like that. That code has to be given here. And other any non-proprietary names or any national names, any US United adopted any names, or maybe Japanese name, British names has to be provided. There is a chemical abstract service number. It will be universally registered one. So if you put one product name, when the chemical abstract number, there will be one number. That number has to be provided here. So this is the product already registered in the market. Similarly, structure. If you take the structure point of view, so the product, what is the structure? If you take monoclonal antibody, there are heavy chain and light chain with the Y shape which connect with the 2 heavy chain and light chains, and having a connection with interdicellular bonds and form a one monoclonal antibody structure. When you're talking about insulin molecules, there is a chain and B chain connected together a dicellular bonds. That required to mention here. So part of biotech, the schematic amino acid sequence required to know, and indicating glycosylation sites required to know. Other post-translational modification related to molecular mass should be required to know. Like this, structurally should be aware of your respective structure of the product. That means technically we should aware the person who is going to write this dossier for the part of dossier submissions, dossier preparation. The person should be aware structurally what is this kind of molecules. So that is what we are going to learn from our sessions. Then general properties. So what is the general properties of this molecule? Like physicochemical properties. With respect to product, we have to provide it. There is a reference guideline is given, Q6B with specification. Very clearly can refer biotechnology products. What are the specification limits? Then coming to manufacturers. This is a address. We will discuss later this one. Regarding part of test license, we will get this address with manufacturing license. That address is going to provide here. Then description of the manufacturing process. If you remember, day 1, we explained about respect to QBD, quality by design. That quality by design, again, we are going to discuss around 4 to 5 days and going to discuss about quality by design concept with respect to biological product. In this case, batch scale definition required to know, and the upstream process with respect to 3 case studies like insulins and respect to insulin products, MABs, vaccines. We'll take up 3 products as a case studies. Then complete upstream process will be discussed here. Then purification. Those 3 products only continue downstream purification. So we know that how we are going to purify it, how we are going to remove the process-related impurities, product-related impurities, viral impurities, that will be covered here. Then filling and storage of your prepared drug substance, how we are going to fill it in the containers, which containers you are using. Is it required any extractable, leachable reports, leachable studies, storage conditions. Are you storing at 2 to 8? Are you storing at -20? What temperature we are storing it? And temperature, what temperature you are storing it? Is there any transportation or any shipping validation is required? These are all part of our studies. Part of our, there is a session regarding transport validation also. There is a separate session like this required. Then control of materials. So part of cell banks will be covered here. Is a key process materials, key input materials, raw materials which you are using for the manufacturing, cell banks, everything going to discuss here. Similarly, complete information regarding cell banks constructions, cell bank characterizations, cell bank preparations, cell bank issuance, cell bank annual stability studies, everything is required. That is going to discuss coming 2 to 3 days. Coming 4 sessions will be complete regarding input materials. Part of cell banks will be major study. And here going to refer all Q5 guidelines. So which is biotechnology guidelines with viral clearance, cell substrates, and your stability and your characterization all going to refer here. Then control of in-process specification. It is part of our QBD and the specification what you created that has to be followed. Process validation. So that is the reason we given a demonstration and part of our regulation also. One more time we are going to review our process validation. Then manufacturing development. Nothing but R&D development till commercial phase, how your process has been developed. So there is a process development report in the R&D. We should collect this document. We'll go for development of this section. Similarly, protein characterization. There is a structural elucidation as well as impurity characterization. This is also having separate analytical section. There we'll discuss in detail. Then control of drug substance, specifications, analytical procedures, validation. So if you remember, one of the session we given validation. Again, we are going to discuss regarding how to do validation, and the discussion of completion, we are going to write those here. And reference guidelines also given here, very clear. And JOS, batch analysis, reference standards. This is also one of the session going forward. We want to discuss regarding reference standards. Container closure with respect to DS. What container is going to use it for the storage and what is the studies is required. Similarly, stability point of view, very important for the stability studies. Any registration, any molecule requires 3 stages of stability: R&D, PCT studies, and clinical studies, clinical batches, and commercial batches, regular commercial batches. So the studies required accelerated condition stability, nothing but formal stability study, and long-term studies, as well as post-degradation studies like photo stability, thermal stress, mixing studies, low pH, high pH, everything is required. So that to develop this type of dossier, we should know very clearly regarding our drug substance strategies. So this dossier, whatever required to develop, the technical sessions coming 20-25 sessions going to discuss same thing. Hope it is clear to everybody.
Yeah.
Now we'll end the session for the day. So when it is going to drug product, that time we'll refer it. Similarly, how we having a drug substance, similarly having a drug product also 8 sections. From that there is having a R&D development, and there is a manufacturing and related to excipients, and related to analytical part, and related to reference standards, and related to container closed system with the DP. It may be vials, it may be pre-filled syringes, cartridges, and related to stability. Appendices, like there is a with regional information, with reference product, regional information with region specific requirement. Like this, if you study for 40 days, this will be cover complete model 3. So when you model 3 study, we'll go for those development. Yeah.
So one more thing I wanted to ask before we move ahead with the today's session,
please.
which is like while we say there is a requirement of CAS number, but while we dossier development, we go for the active substance or any drug product also, even in Europe or US or Canada, which we have done. Even for SED, there's the same structure that we follow normally as per the requirements. So do we have any kind of such number for biologics also specifically mentioned? Somewhere?
Yes. If you see our dossier, for example, I will show another day. It is time already up. It is number, that is a fixed number. Cost number is a fix. If you go and search it,
even for biologics or biosimilars, it is there. Then I think that's the only concern that I wanted, because
same.
Okay.
If you go on internet, if you simply Google it, cost number for insulin glargine, that is the universal standard. Where that cost number using at Biocon, using by Intas, using by Redis, the number is a standard, fixed universal standard.
Okay, okay, sir.
Yeah.
Thank
So
you.
this is the guidelines part of our discussions. Going to discuss all these guidelines going forward. We're having each and every guideline also. We'll refer wherever is necessary. We'll refer it, and you need not to study. You focus our session for 90 hours. You will get it what is there in this guidelines, and you are based on your interest. You can go ahead further also to review and referring these guidelines also.
Okay,
Yeah,
sir.
yeah.
Thank
That's
you.
all regarding this overview of the session for coming 3 months. What you are going to do it. So anyhow, regarding drug substance, I given a clarity. Similarly, when you go for drug product, there we'll discuss regarding drug product. Yeah.
Thank you.
Yeah. Yeah. Any questions? Anything? We'll start now one by one. Yeah. Please stay back. The people wish to clarify anything, or it is already clarified. Then you can stay back for any other questions from other end.
Well, I would like you to clarify the interchangeable biosimilars and biosimilars maybe another time. I didn't get that side quite well.
No worry. It will see the people who is not background of biosimilars. It maybe looks like somewhat difference. We'll get it more going forward. Every day we'll go and discuss about the biosimilars, biologics things. So we'll get more clarity on it.
Okay. Okay.
Yeah. So with the session feedback this Monday regarding the other part.
Yeah, very good section. Yeah,
Okay.
in fact, everything relates to. I've seen most of the things that I do. I mean, being presented on the on this training, and I'm beginning to understand it better.
Okay, great. That's what we want. Yeah, Dayananda.
Yes.
Dayanand?
Yes.
Yeah. Any questions from your end and with the session feedback, understanding of the session?
Yeah, it's clearly understood. As of now, there's no question. If any, I will come personally, please.
Okay. Okay, fine. Madam Hema?
No questions from my side. So I understood how the flow of the coursework would be for the next 60 class. Thank you.
Correct. Yeah. Dr. Ketana?
Yeah, the course is good. Since I'm very new to this regulatory affairs, my thing is how could where should we find these guidelines? You said this Q6A, this Q5. Is there a general document that tells us about all these guidelines?
Yes. So when you're going for developing any manufacturing process, so that the manufacturers used to adopt these guidelines, and he has to take a reference of this guideline. And when you're going to use this reference as a guideline, what guideline is saying, accordingly, made the is explained. And when you're going for any evidence, we are giving as a reference of that guideline, so that when it's going for submission, so the agency is asking any query, so we can give it this reference guideline. This is the guideline saying, so we followed. So that will be having a supporting guideline and reference guideline will give more supporting data for the filing point of view.
So this is a universal one.
Universal one. ICH is the standard guidelines, can be used worldwide, anyone.
And you can find this in the internet. So
Internet, yes. It is simply if you Google it, it will be happen. At the same time, I will also keep on sending the reference guidelines wherever we use for the particular day. I will send through your emails so that you can download or you can save it in your systems. It will be available internet. Everything is available internet. Only the search option you have to use it.
Okay. And when we write, you showed last the document, right? Where for the quality, you write all these technical document.
Correct.
So is this like in the company will already have this fixed document where everything
available. Everything
okay.
Will be as a source documents. For example, you are going to write the manufacturing development section, or maybe your ease of understanding, cell bank preparation document. There is a clone development document already available in the molecular biology department, and already someone is reviewed and someone is already approved by quality assurance department, and it was available in the quality assurance department. When you're going to develop those section, and the first point of view, we may be required to refer 10 different documents.
Okay.
And by referring 10 different documents, we have to make one dossier section. Is a scientific write-up.
Okay.
And in general, you may hear that word medical writers. Similarly, here, in generally we'll call regulatory sciences persons will start writing the 10 different documents by referring. We will write it. So we should refer that guideline, M4Q guideline, what guideline is saying. Accordingly, we start write up of your document, those document. That way we can write it. Yeah, we will take one person help. Madam Roja.
Okay.
Roja, are you there?
Yes, sir. Can you hear me?
Yeah, you are developing the doses, right now?
Yes, sir. Yeah.
So what section you are developing in generally?
Mostly for drug substance, all the process development sections,
Okay.
like
For example,
manufacture.
correct. For example, you are going to write section number S2.4, which is critical control of critical steps.
Critical steps, yeah.
Which document you are referring as a source document for it?
So we will have a control strategy document which talks about all the thing and specifications also.
Correct. In process specification.
In process specifications. And then we'll have the analytical method. So how we
We'll give
do
a reference. Yeah, we'll give a reference to the.
Yeah. And if required, a description of that method, if there's any difference in that.
Correct, correct. So, so by referring those documents, you are going to prepare a dossier section.
Yes, yeah.
Correct.
And
This is the
we'll
way.
write the whole time studies, whole time data.
Correct, correct. That is part of the strategy we can put it.
Yeah.
Yeah. Got it, Kirtana, Dr. Kirtana?
Yeah. Now I'm coming into it, but maybe
it's 90 hours.
I will learn.
Yeah.
Okay. Thank you.
90 hours dedicatedly attend it and dedicatedly watch the, if it's not possible, also physical class, online class. You will get it. Nothing to worry. This is the day 0, where it is interesting, quite interesting. Every day we'll learn new, and we'll reach to the requirements one day.
Sure. Thank you.
Yeah. With the other thing, Dr. Kirtana's session understanding, is it fine? All things?
Yeah, it's fine. I'm clear with the idea. Means background is okay, because this how to enter into this regulatory documentation is what is new to me. So that is what I'm learning.
That will come back. That will come back. By understanding technical things, that is pretty easy to write the regulatory documents.
Sure. Thank you so much.
Yeah. Madam Lakshmi. Okay. Roja, with the session feedback? Yes, madam. Any doubts, any questions, madam? Lakshmi, madam?
Yes. As I said, like we'll discuss it later. I'm having a doubt at the what we say that
interchangeable biosimilar.
Yes. Interchangeable biosimilar.
Just stay back, madam. I will ask other questions. Anyone is there, then we'll come back to you. Yeah, I'll open again that.
Sure.
Shrestha Verma.
I have no questions.
So how is the understanding session?
It was okay for me.
Yeah. Okay. Thank you. And C, how we can call? Can you pronunciate? From Jumambe, Joan, thanks for joining. C, are you there? Okay. Anyone else you want to ask any questions? Dr. Virendra?
Yeah. Regarding my one question, sir. As you told in sheet 3D, and it said the kind of papers in each module like 2000, 164. Overall, how much maximum pages CTD, entire module 1 to module 5?
See, there will be no limitation to the number of pages based on the
ideal. Like you told 2000 for module 3, and so if you give the overall to understand the non-person to understand maximum how many pages.
See, there is no any fixed pages, but especially for biologics.
With respect to drug substance, with respect to biologicals, so approximate rough idea, like you told 2000.
Rough idea, you can consider around 2000 pages for the drug substance, and maybe some 1000 to 1000 pages for the drug product. Another related appendix section and regarding our biosimilarity may come around 500 to 1000 pages.
So an in consider all 5 module, more than 5000 pages will be
it may reach to 10,000 also.
Okay, just I want to understand the broad range.
Yeah, because
and what about ECTD?
ECTD is nothing but just you are using a software to upload the prepared CTD. That is that is not
matter should be less, shortcut now compared to ICS submission. As you told, 10,000 pages for any highly regulated market. So if you want to go for ACTD, what are the proportionally cut down in the page number, like 10,000 to rough idea?
ACTD for only for Asian countries. There are 10 countries. it will come for only ACTD structure format for the only Asian countries. There are 10 countries.
Yeah.
When coming to ECD is one of the software, it is one of the app. We are going to, instead of submitting the manual documentation, we are uploading in the, for example, you are going for any applying for the job opportunity. 2 ways will be there. One thing, you can write an email to the company by attaching your CV. Another thing, you can print or maybe you can take any electronic submission of document to the giving directly the application or maybe your CV to the directly to the HR department. 2 ways is there. Where which one is the easy for you? Is it a physical copy submission in the HR department easy, or sending an email through attachment easy? Which one is easy?
Electronic is email.
Email is easy, right? So you're having evidence and you will ensure that the document is when it is submitted and we can track it also. So similarly, the dossier submission which is prepared CTD dossiers, there are 2 options. One, we can through OneDrive access we can give it, or maybe a CD or copy thing, something we can share it. Other thing,
and manual based, electronic based. Only my simple question, sir. As
so
you
there is nothing reduction of pages. What you talking about ECD? Nothing reduction of pages. Only you are uploading through ECD format, electronic format through some licensed app-based service software.
Okay. So the kind of content and pages will be same. The mode of submission and the kind of structure of pyramid differs. That's it.
Exactly.
Yeah. As you told, we viewed the module 2. Like there is no module 2 in the particular ACTD Asian country. But the content will be same. Okay. Yeah. Fine. Last one question.
Yeah, please.
When you compare the molecular mass of different molecule from small molecule to large molecule, so you give an example of aspirin. So can you show that, sir, content of PPT? The molecular weight of aspirin shown there 180 Dalton.
The presentation slide is visible?
Yeah, that's when you compare the different molecule to study the case study. Yeah, this slide. Yeah, first molecule, small molecule aspirin.
Correct. This is the aspirin, which is 180 Daltons.
But ideally, sir, we'll mention small molecule. We need grams per mole, actually, when we understand.
No, but when you go into chemical mass, when you're going to extract, it will come in the Daltons. We can able to calculate. Like carbon is there. How many? So what is the mass?
I want to understand Dalton to gram per mole. How any correlation or any? As you comparing this small molecule.
That one, that will discuss separately. That will discuss separately.
That bit. Okay, thank you.
Yeah. How is the session, Virender, for your understanding?
Yeah, it's good. The beauty of presentation is that you are showing each guideline and ensuring the section that makes more practical and aware the thing.
Okay, fine. Yeah. So
and
now,
last one question, as you given in 3.2 yes, 3.2 P and 3.2 A also mentioned. What indicate A segment?
A indicates appendices regarding your facilities and equipments.
Yeah, amendments are this appendix.
Equipments and regarding facilities, like what type of classification of areas you have. With the GMP facilities there for man entry, exit, material entry, exit. Those are all
outline of the facility outline, like how you told in city administration.
If you see our syllabus, and there is a GMP class under area classification, also one of the session is there. So there we'll discuss regarding that one. So whatever 60 sessions is there, entire those are requirement only.
Fine. Okay, thank you.
Yeah, fine. Lakshmi? Madam Lakshmi, are you there? Oh, she left. Okay. Okay. Is all clear at everyone? There is a messages. Anyone is there? Okay. Can you give one latest example of biological and biosimilars? Already clarified, Virender, this one. Yeah. Okay. So there is no more questions. Yeah, we will stop the recording and we will end the session for the day. We'll connect.